Hello everybody. I don't know if you can hear us, but I think yes. Perfect. So we are very happy to organize the last webinar of the RFMF today, last for the year 2024, and it's a great pleasure for us to have Dr. Laura Sanchez from University California Santa Cruz to speak about the Holobiont concept, one subject that we want to pinpoint for the last webinar. So she is associate professor since 2015, first in the University of Illinois, at Chicago, and then she moved to University of California Santa Cruz to continue his research. And today she's going to speak about cheese microbiome and as a model to study microbiome and bacterial-fungal interaction. So Laura, the floor is yours.
Okay. It's great to be here this morning or for you all in the afternoon, and we'll just go ahead and get started. So I always like to acknowledge the people that do the work. So I'll be telling you about work that Jess and Gordon started and that Carly, a postdoc in the lab, is currently working on. So let's go ahead and get into it. I think we can all, let me see, okay. I think we can all appreciate that, microbiomes are essential for all forms of life, whether that's plants, people, or animals. And that when I say microbiome, we're really talking about communities of microorganisms. And I'd also like to highlight that, these microbiomes are obviously more than just bacteria. So I think bacteria have really taken as a natural product chemist, we like bacteria. They've made a lot of things, but these microbiomes are actually they're more complex than just bacteria. And the reason we like cheese is that they're both, the cheese rind itself is bacteria, filamentous fungi, and yeast. So this work started in collaboration with Rachel Dutton and Ben Wolf, whose labs described the cheese microbiome. So they took inventory of a lot of different cheese rinds from across the world. And the other thing we like about this is that it's really consistent. So we know, and kind of a harsh environment. So, at the start of the cultivation of these rinds, which you can see here, we know what the salt content and the pH are. And then over time, we know that there are these trends in bacteria and fungal growth patterns, accompanying the fact that the salt content goes down and the pH rises. And then from a chemistry point of view, if we look at a similar kind of cheese aging over a 2-month period, we can obviously observe that there are morphological changes and color changes occurring on these rinds. And as a chemist, a clear sign of a chemical reaction is that, color change. And so when we started this project, we were really interested in understanding what kind of chemistry accompanied these aging microbiomes, and why are the cheese rinds so consistent, and how are these fungal and bacterial growth patterns the same time and time again on these artisanal cheeses. And when we started, there wasn't a ton of places to start, so originally we'd kind of taken this pairwise approach. And so Rachel's lab, we were using cheese curd agar, which is, I think, the other really interesting thing about this system, is we're giving these microbes exactly what they want and need and would replicate in their native environment. I know some people like to push back and say this is not a native environment because it's all totally man-made, but okay, whatever. So we actually use cheese curd agar. And, at a very simple level, again, when we grow these bacteria and fungi, so the penicillium in this case, on the cheese curd agar, sometimes they don't grow well alone, but when you pair them together, you obviously, again, get these color changes. And we see these, again, on these bigger petri dishes, instead of just these 96 well plates, that there are growth phenotypes when we grow these things together. And again, the presence of this pink pigment we started with, because this is occasionally observed on cheeses, and no one knows if it's good or bad. So we're like, okay, why do we have sometimes pink cheese? And is that related to the interaction of the Glutamicibacter and the Penicillium. And again, this is a very spatially driven question because these things grow on surfaces. So I want to introduce the concept of imaging mass spectrometry, which is something that my lab is really our hypothesis generating tool. So hopefully the video is playing. And what you'll see is that we grow bacteria and fungi on these thin Petri dishes. And when I say thin, I mean this is about 10 milliliters of agar until we have the phenotype that we're going for. And then we just physically excise this. So in my lab, we use MALDI, which is matrix-assisted laser desorption ionization, as our form of imaging. There are many other forms and ways to do this, but we like MALDI because it is fast, so it's laser-based. So we can, irradiate the samples with, high frequencies. And it has a good spatial resolution. Although these things grow so big, we don't necessarily access that spatial resolution capability for these samples. But anyway, so we take this thing, put it on a MALDI target plate, and those target plates are about this big. So that gives you a frame of reference for the scale at which we can do these. Then we put our target plate into a sieve. So these are 53 micron stainless steel sieves and coat it with a matrix. So for this form of mass spectrometry, because it looked like a lot of the other speakers were going to be talking about LCMS-based metabolomics. MALDI imaging is really interesting because it's a solid sample. So we're going to coat our solid sample with this matrix, which is small organic acids that are really good at absorbing energy when we irradiate them with a laser. And we need this matrix to co-crystallize with our analyte. So you'll see once this plate comes out that has a bacterium on it, it has a nice even surface. And we really only coat these things for a couple minutes. Undergraduates can do this. It's very straightforward. And then we desiccate the entire sample because this does occur in a vacuum and we don't want the sample to explode. So kind of like cooking, we bake this thing just at like 37 degrees for 4 hours. That allows the matrix to co-crystallize and also essentially adheres our sample to these target plates before it goes into the instrument. We then irradiate across the sample at a set spatial resolution. So you can see that by these little red dots, that would be where we've measured. And at each single point, we collect a full mass spectrum. So originally the images I'll be showing you were done on a TOF, so a time of flight. So we were able to go from like 200 Daltons all the way up to 2000 and really take a look at what kind of chemistry is there across the sample. And then in the software, it takes an overall computational average, and that's where the fun comes in for the imaging experiments. So we're able to look at specific mass to charge ratios or ions and where they were with their intensity across the sample. So we map them back to these original raster points. So we can see things that would, for instance, be colony associated, colony excreted, or just everywhere. So that's either the cheese curd agar or the matrix itself. And this allows us to generate that hypothesis. So if you recall, in this original interaction we were going for, we're really interested in why does the peak pigment occur when we're looking at a penicillium growing with Glutamicibacter. And so what Jess did originally was to grow these things together and then alone. And we were looking for ions that turn on only in the presence of the co-culture. And so there weren't actually a ton of candidates here. And I'm going to summarize a lot of work that she did. But like long story short, we were able to prove that this ion is coproporphyrin 3. And it is specifically 3, not 1, 2, or 4. And then furthermore, that it's actually zinc bound. And we were losing the chelation with zinc in these imaging experiments because the matrix itself is slightly acidic. So in this interaction, specifically, zinc coporphyrin 3 is produced. And so then in order to validate this, we worked with our collaborator, Ben Wolf, and he actually ended up looking at the transcriptomics. And when we look at Glutamicibacter alone versus Glutamicibacter with penicillium, we can see that the entire pathway for the synthesis of this molecule is upregulated when it's in co-culture with penicillium. So this is great. Like this appears to be necessary for their interaction together. And then furthermore, when we test these things to see if there's any growth defects, we find that it seems at higher concentrations to inhibit the growth of the penicillium species. So we think that this is an example of these molecules that are really important for how microbes and fungi grow together. But this was just one example. So the next thing we wanted to do was really like how widespread are these interactions, and how do we try to test this? So we thought it would be good with Rachel's lab to try a paired metabolomics transcriptomics experiment. So we took 8 of our favorite fungi from the system, so different penicillium, a fusarium, some scapulariopsis, and then some yeast species, and paired those against 2 different bacteria that were genetically tractable. So we looked at E. coli K12 as like a model invader for the system, and then Pseudomonas cyprophila, which is a normal strain that is supposed to be on these cheese rinds. And as you can see by eye, when we look at these pictures, there are obviously different phenotypes depending on which bacteria and fungi we pair together. So it looks like the commensal to me almost always grows better with the fungi compared to the E. coli, which sometimes is just dead or not there. But now that we have these phenotypes, we wanted to know what was actually happening. And the first thing we did was to actually look at the transcriptomic data. And Emily, a graduate student at the time in Rachel's lab, had done barcoded transposon insertion libraries to look at fitness. And I'm going to summarize like years of her work by just saying, long story short, we found that bacteria largely had a couple of conserved responses to the fungi that they were paired with, and it didn't matter which fungi. So largely bacteria all experienced cell wall stress, and that siderophore requirements for the bacteria were altered anytime they were paired with these fungi. And we did find that there were a couple like unique interactions out of the system, but largely these were the ones that stood out to us the most. And so we were like, okay, what about the metabolites here? Can we figure out what fungal metabolites are responsible for these effects that we were seeing? And so to vignette this, Jess did a lot of work. So we grew monocultures, co-cultures. We did these things in biological triplicate. We did the imaging, mass spectrometry, but what I'll focus on for this talk is the LCMS and the LCMS-MS using ESI metabolomics for these studies, because we were interested in understanding both the quantity and the identity of the molecules. And so that's where we started for this. So the first thing that I want to point out on this slide is that anytime I'm talking about these, the little different fungal species will be up in the corner because sometimes I'm not going to be talking about all of them. And we've tried to keep the color coding consistent. So all the penicilliums are shades of green, if you will. The scapulariopsis are always going to be these yellow-orange, and then the yeast are going to be the blue colors. And so the other thing I want to orient people to in this is that the first thing Jess did was just to take an overall view using LCMS, and this was done in MetaboAnalyst. So how do the fungal metabolic profiles change when you have E. coli versus growth with Pseudomonas versus growth alone? And largely what we found when you look at these, the hierarchical clustering, so the conditions are on the top, the features are on the y-axis, that largely the fungal species do cluster with one another, which is great to see. So all the penicilliums are together. But we found that the fungal profiles were largely unaffected by their bacterial growth partners. And that the other thing we noticed kind of right away was that the yeast are a little bit boring. So nothing really seemed to change much. So maybe this wasn't too surprising if you work on fungi. They just have so much more biomass, and I think their genomes are just gigantic comparatively. So we were like, okay, the fungi don't appear to care who they're growing with in this instance. But what are these metabolites or ions or features? So we'd like to use tandem mass spectrometry to try to get at this information. And I think everyone on this call probably knows that that is, but just in case there's like a brand new graduate student, we like tandem mass spectrometry because we can take a molecule. I like to think about these things as puzzles. We apply energy and break them into our puzzle pieces. And I am sure there are some people on this call that are like master puzzlers that can fully solve molecules based on spectra alone. And there are others that computer programs are like pretty good at this nowadays. And the program that we like to use is the Global Natural Products Social Molecular Networking. So I worked on this when I was a postdoc in Peter Dorstein's lab. And for those, just to orient you to what you'll be seeing in a little bit, we're going to be looking at networks with nodes and edges. So a node is going to be labeled as a mass to charge ratio, but it is actually the MS data for these compounds. And multiple spectra can go into single nodes. So the first step in the algorithm behind the GNPS platform is that it looks for consensus nodes first. So it tries to find like spectra out of your data, because if you're going to upload 78 data files, you kind of want to make it efficient while it's doing this. So the first thing is it looks for consensus among consensus spectra to create a consensus node. And then it's going to be connecting nodes of your MS data using a cosine similarity score. So a cosine score of 1 would mean it was identical. The lower the cosine score is, or the closer it is to 0, then it doesn't believe that they're identical at all. So those would be probably unrelated. And I think we can all appreciate that compounds from similar families likely have similar fragmentation patterns. So we hope we can seed these networks with like libraries of compounds to try to make sense of the unknown, known unknowns or the unknown unknowns from the network, to get a feel for what we're dealing with. So that's the first thing Jess and Gordon did. And from, a view, these networks sometimes I find they're just like a little bit overwhelming, but what we can see are things that we expect. So we found peptides, fatty acids, lipopeptides, polyketides. So kind of like for a natural product chemist, it's everything you would probably find if these had been soil fungi. So we found the whole range of everything. And when we further broke down just the known compounds then, using a variety of methods, what we could see is most of the identified chemistry was coming from the Penicillium species, followed by the Scapulariopsis. So these filamentous fungi were major contributors of known compounds to these experiments, which was nice because then we could figure out what they are. And again, the yeast remain a little bit unexciting. And so, yeah, the filamentous fungi are the major producers of the natural product-like compounds out of the system. And again, I'm kind of summarizing a lot of work, but the fungi do produce bioactive compounds. And then we found that all fungal species produced at least one, if not multiple siderophores. And so we were like, okay, why does siderophores or these iron chelators keep coming up? So one experiment that Emily did to test this from Rachel's lab was to grow these same fungal species. And then what you're looking at are panels of bacteria. So we have our wild type E. coli, and then she made mutants of the E. coli's siderophore uptake and transport systems. So what you can see here when and then these E. coli, there's something in the cheese curd agar to make the E. coli turn red just so we can see it. Otherwise, a pale bacterium on this kind of agar just doesn't show up. Oops. And so what you can kind of see is that, when E. coli is intact, it's growing just fine, especially if you look at this top row. And then in other instances, when various transporters have been knocked out with, single or double mutants, we can see that the E. coli can still grow in some instances. So even though it theoretically can't uptake or transport its own siderophores, it's able to essentially be, like I've decided bacteria are lazy cheaters, so it's able to use the siderophores that all of these fungi are producing, which I think explains the transcriptomics data that we were seeing, that all these fungal species have the capacity to make siderophores. So I think this is really interesting, and I think this also explains why the bacteria are more responsive to the fungi than the fungi are to the bacteria. And so, yeah, we went on, and I don't have time to talk about everything. I wish I did, but we went on to classify all these siderophores out of this system. And so a lesson that we learned originally when we took this omics approach was that, the metabolomics of multiple species is not straightforward. We ended up using, instead of just cutting things out, we now use a cookie cutter approach. And by cookie cutter, I mean we use a 50 milliliter Falcon tube, and we found that has really added a lot of consistency to our experiments that we did not start with, because I will say Jess and Gordon did this a couple times. And that I think everyone probably appreciates that The sum of your metabolomics experiments are much greater than individual experiments alone. So I think both pairs of data were able to contextualize one another. But I think one of the things that still strike us about this system is that there's a lot of functional redundancy and synergy of the metabolites that we did see. And so we wanted to try to figure out if there's a way that we can more systematically test this, because I think, as these experiments get bigger and we have more and more data, it becomes harder to figure out how we design, really rigorous experiments. So the hot off the bench data and what we're planning on doing now with the same system, since we believe that fungi are really potentially the drivers of the system, or at least very important for the structure of this community, is to try to impact their secondary metabolome, because they were really resistant to whatever we put next to them. So the plan is to pick a specific global regulator in all of our fungi and knock it out and see if the fungi are lacking now some of these essential natural product production capabilities, what happens to that entire community. So that's how we're going to try to test this system in this next iteration based on the data that we have at hand. And so just to give you a flavor, so one of the fungal species that does have a global regulator that we're going to be targeting is this Aspergillus species. And this is actually an example of a fungal invader. So this is what it looks like. So there's been a semi-recent outbreak of this in cheese caves, like in Vermont here. And what you can kind of see that I thought was fascinating about this Aspergillus species is it's almost like killing the rind. So this to me almost looks like a gigantic petri dish. So what you can kind of see is that the rind is almost kind of bald where the Aspergillus has grown. And again, this is very spatially driven. So we're really interested in understanding, what kind of metabolites is Aspergillus making, and then if we knock down one of its global regulators, how does that impact the system? So again, this is kind of hot off the bench. So we're starting with what does this fungi even make? And to accompany this, we do know when we look at these communities, so we're going to again be using our Psychobacter, a Brachybacterium, and a Brachybacterium, and a staph, and these are all natural rind cheese microbe community members. We found that this effect of Aspergillus killing the community is very temperature dependent. So you're looking at 10, 15, and 20. And you can see at 20 degrees C, the community, when we add Aspergillus, is the colony forming units of the community are all decreased compared to when the community is just growing alone. But that this does essentially trend back to normal if the cheese is cold enough. And so we're really interested in understanding, what metabolites are related to this, and do those spatial distributions have anything to do with this? And so the way we set this experiment up now, and what we're doing these days, is instead of growing kind of one by one, we have the fungi on one side, and then we're spotting the bacteria on the other. So you can see that these things usually take 7 days. We've grown these all the way out to 2 weeks as well. And you can tell already that, the phenotype of the bacteria is impacted by the fungal growth. And I don't have a picture of this in this presentation, but when you leave this even longer, you can almost see the fungal spores essentially colony jumping, which I find really interesting. To me, it's almost behaving like it's a mix of bacteria, which I've never seen before, so I thought that was kind of cool. And so Carly was a brand new postdoc when we started this project in the lab, and she was really excited to learn how to do the imaging. So I just want to give you a feel for what her data looked like when she started this. So you can see we have a really complex, MS1 spectrum. So this is the total composite out of the data set, or that master spectrum that I mentioned at the start of this talk. And the way we set this one up is you'll be seeing your controls up here. So we have the Aspergillus grown alone, the Brachybacterium grown alone in the agar. This is the dried optical image in this case. And then the regions of interest for the different bacteria spots were essentially imaged on their own to try to do a little more statistics behind the scene. But anywho, so like this would be a signal where we would again attribute it to the media or the matrix because it's high abundant in every single condition. Something like this we would attribute to the Brachybacterium because we can probably say that the colony closest to the fungi is on its way to dying, if not dead already. So we can see that in these colonies that are further apart from the fungal species that we're getting high intensity of whatever this mass to charge ratio is, and that maybe this is also upregulated when the bacteria are grown together, or maybe it's upregulated in the presence of the fungi because we do see it in the brachy grown alone. But that's not necessarily indicating to us, what might be impacting the bacterial growth. Something like this may be a candidate. So we see that it's something that Aspergillus makes when it's grown alone. And we can also see it radiating out of the colony as well, headed towards the bacteria. So that's great. And then there are other things that we're also kind of very interested in. So there are signals that are only appearing in the co-culture. So like this would be one where it's just between these two bacteria colonies and not in any of these. And then another example where maybe in this case, the fungi has actually turned something on that's unique. But in the end, you're probably like, oh my God, Laura, what do you do with all this data? And that is exactly what Carly said. So it's a lot of data. And so the nice thing with this interaction to get started was how do we prioritize all these signals? Because I'm just showing you a small subset of how rich this data set actually is. So Carly turned to genome mining. So the nice thing, again, about some of these organisms that has occurred over time is that Rachel and Ben have sequenced quite a few of them. And when we started this project, we did not have the genome. So that, kind of made everything a little more complicated. So what we were hoping for was to kind of pair, okay, what is expected versus like what is actually in our data set. So one of the things that Carly was able to find that struck her was the presence of ochratoxin B. So this is one of the mycotoxins that we know that Aspergillus should be making. It's a reported ochratoxin A producer, but we never found ochratoxin A. And ochratoxin A is chlorinated, so we should be able to select that, that has a really nice distinct pattern. Specifically, we've never found it. We've only found ochratoxin B. And when we went back and looked at the imaging data, it is at this interface. So we're pretty interested in following up on that. And then the other thing that struck Carly when she did the molecular networking, which was again to kind of take these things and look at the LCMS, because the other candidates we found via genome mining did not appear to have biological activities per se. She found a hit in the GNPS database. So again, this is a butterfly plot. This is her experimental spectrum with a standard that was in the database, was potentially penicillic acid, which is another known mycotoxin, but the biosynthetic gene cluster for penicillic acid is not known. So that kind of explained why she didn't see it in that data. And when we went back and looked at this, there is a little bit of a dead spot here. This is a replicate now, but what you can kind of see is the signal is actually radiating out. And I will also add that in our imaging data, she found both what appears to be M plus H and M minus water, which is a little bit odd. But luckily, this compound you can commercially buy. So we're interested in understanding, whether these two things might be interacting. Oh, and I will say it appears to increase over time as well. So there is one paper that reports in a mouse model of infection that there's synergistic activity between penicillic acid and ochratoxin A. So we're interested in testing whether ochratoxin B might also have synergistic activity with penicillic acid against these different community members individually and as a bacterial community. So that's something we're in the process of testing with Ben right now. And the other compound she found that I didn't think I had time to talk about, which I don't think I did, were the ochra indoles. So these ones were kind of produced in all the co-cultures, but I think this highlights, how important it is to kind of pair the genomics and the metabolomics data or the transcriptomics and the metabolomics data so that you can kind of complete these richer pictures and also subsequently mine these data sets. So we're in the process of testing we just had a rotation student do this, testing the other community members before we put the community back together, and testing the synergistic effects. And so with that, I'd like to thank the people that did the work. So Carly is in charge of this project now, and she took over and learned the lessons from Gordon and Jess, previous PhD students. Rachel started this project with me, but she's now in biotech, and then Ben is at Tufts, and we have been historically supported by an NSF. And so with that, I'd be happy to take any questions. And this is the lab. In case you're like, what are you doing in the United States? We're all dressed as Olympians. So I'd be happy to take any questions. And I might stop. Oh, I might stop. Okay, I stopped sharing. Okay.
Yeah, you stopped sharing. Thank you very much, Laura. It was very interesting. So please don't hesitate to ask a question in the chat, or if you actually prefer to ask your question directly, please don't hesitate to unmute your mic as well. So far, we don't have any question in the chat, but Samuel, do you have some? Or I have a small one. So I don't know if you want to start, Samuel. I'm pretty sure you have some in the metabolic network.
Yes, meanwhile, people can have also questions. You started some experiment with a large, diverse mixture with a lot of different microorganisms. Did you see any convergent selection of only a few at some point that you can that came out to be the stable community from a complex one to a simpler one with some time? With cheese?
Yeah. We have, so basically for the stable community, we almost always use like a Staphylococcus species with one of the two actinomycetes and then the Pseudomonas. And we think that kind of represents, like that grows just like it does when we've compared the metagenomics on the cheese rinds. We can recapitulate that in about 3 weeks in the lab. So we've kind of landed on that being our really stable go-to community for natural rind cheeses.
Mm-hmm.
Some of the other ones, so yes, there are specific smaller communities that we can make now. Instead of using, I don't think I said this, 10 to 12 microbes per rind, we're kind of down to this like 3 to 4 bacteria in a fungal species, replicates what we see on the cheese. And it also follows with like the aging too. So when we have these specific communities, we see the cheese curd agar, turn brown over time, which really mirrors what we're seeing on the cheese. So we think that represents a nice minimal community.
Okay. Does anyone have?
I think I see someone who is raising his hand.
Yes, hello. Hi, Laura. Thanks for the nice presentation. A quick question. Maybe I missed something at the end, but so you say you can use genomes to link metabolite prediction from what you see from the metabolomics part, but then you said that for one of the last compounds, there was no signal for no reference in MyBig, no hit with anti-smash. So can you be a bit more specific on this? How did you do to link it?
Yeah, so in that case, so Carly was basically using anything she could to try to annotate the imaging data. And so that one she just put, so in addition to like mining the genome, we also have gathered the LCMS-MS data. And so that one, we still don't have any idea about, we don't know what its cluster is, but it does come out, and we've been able to now retention time match it. So it is something that the fungi makes, but we don't know what the gene cluster is.
Okay, thanks.
Great. And I have a small question about, I've seen the annotation that you've been doing on with imaging. I was just out of curiosity. I have no idea how you do that. I know that in not imaging, what we do to go from annotation to identification is usually spiking experiment, where you spike your sample with the standard of the putative annotation that you've done. Is it the same in imaging, or is there any difficulties? I have some difficulties to know exactly how to imagine how you can do identification with imaging?
Yeah, that's a great question. So the original data that I started with, we collected on a axial MALDI-TOF. So in that instance, it really was only able to gather MS1. And we always fell back on having to do extracts and, try to mesh the ions as best we could between the ESI-based and MALDI. And for the coproporphyrin 3, we were able to get a standard. We did retention time matching, co-injections to confirm that. But I will freely admit it was a lot harder. And the instrument we're working with now is a Timsestamp Flex, which is a dual ESI MALDI trapped ion mobility, QTOF. And so the nice thing is this has made our lives to some extent a lot easier because we can now gather both collisional cross-section and MS data right off the exact same imaging, as long as the ion is in high abundance. So some of these things we still end up doing extracts and we retention time match when we can for things that are low abundant in the imaging data. So like it's great that this has streamlined some of it, and we do like the collisional cross-section now as another kind of like immutable chemical property. I was originally, I don't know, I'm sure there's ion mobility people. I was originally a little bit skeptical, and I'm like a total convert now. Like the predictions are getting much better. Like there's a whole nother side story where Gordon thought he'd identified a compound, but he's like, oh, the predicted collisional cross-section doesn't match with the experimental, and we ordered the standard and it didn't match. So I was like, okay, like I think the collisional cross-section was telling you it wasn't this compound the whole time. Even though it was close, like it needs to actually match. So we're using that now as like another one of these like immutable chemical properties when we're trying to find higher confidence in our data assignments. So yeah, recommend ion mobility. It's awesome.
Thank you very much. I don't see any other question, and I think it's actually the time to go to the next speaker. So thank you again, Laura, for this great presentation and for accepting our invitation. And we'll not be going too far from Laura research of interest, as we'll be now welcoming Allyson McAtamney, who is a PhD candidate in the lab of Laura, in chemistry and biochemistry, and at the University of California in Santa Cruz. And she will be talking about small molecule communication within a marine invertebrate model, host-microbe system. So thank you very much, Allyson, for accepting our invitation as well, and the floor is yours.
Yeah, thank you so much for having me. Super excited to be here. I am Allyson McAtamney, fourth year PhD candidate in Laura's lab at UC Santa Cruz. And like she just said, I'll be talking about small molecule communication within a marine invertebrate model host microbe system. So the system that I study in the lab is the symbiotic relationship between the Hawaiian bobtail squid and Vibrio fischeri, in which the Vibrio fischeri live inside the squid's light organ and they bioluminesce so that the squid can go hunting at night. So from predators above, this bioluminescence actually cancels out the shadow that's cast by the moonlight, and then from predators below, this bioluminescence actually looks like moonlight. Another really interesting fact about this relationship is that Vibrio fischeri is the sole colonizer of the squid's light organ, and it does this by through biofilm formation. So as the bacteria enter the light organ, they adhere to the surface of the epithelial tissue and produce bacterial biofilms, which are known to assist in host colonization, and they also serve as like a physical protectant for the bacteria, so that they could survive and thrive in that environment. In addition to biofilm formation, we hypothesized that additional small molecule production may contribute to this ability of Vibrio fischeri to solely colonize this light organ of the squid. So with that, I'm going to be talking about two separate projects that I've worked on throughout my time at UC Santa Cruz. The first one is a more targeted project about cyclic diGMP, and then the second part will be a more untargeted project. So with that, I'll go ahead and get started on the cyclic diGMP. So cyclic diGMP is a major biofilm signaling molecule. And once again, this is kind of just an illustration of bacteria adhering to a surface and then continuing to produce this bacterial biofilm. As they adhere to the surface and produce this biofilm, they also produce increasing levels of cyclic diGMP. So it's kind of a secondary messenger, and it is as there's higher levels of cyclic diGMP, there's also high levels of biofilm formation. And this tracks across multiple different organisms that produce biofilm as well. Just talking about the metabolism of cyclic diGMP, there's two main enzymes that I'll be talking about in this presentation. This first one is a diguanylate cyclase. So this is the enzyme that produces cyclic diGMP. And the second one is a phosphodiesterase. So these can either break cyclic diGMP down into PGPG or GMP. So with that, until recently, until our work actually, there was no method to directly detect cyclic diGMP in microbial colonies. A lot of methods previously were using some sort of reporter, which is an indirect detection of these compounds, but using mass spectrometry imaging, we are able to now directly detect cyclic diGMP. So we started by looking at a simple model in Vibrio fischeri, where looking at the wild type, we see that in the microbial colony, there's a decent expression of cyclic diGMP, and when we overexpress a PDE, so the enzyme that breaks down cyclic diGMP, we see a significant decrease in production. And when we use an overexpression of a DGC, so the enzyme that produces cyclic diGMP, we also see an increase in production of cyclic diGMP. So this is kind of a proof of concept that we were able to detect cyclic diGMP and relatively quantitate it as well. Moving forward, we wanted to understand how this looked in Vibrio cholera. So cholera is very closely related to Vibrio fischeri. It's the causative agent of cholera as well. So this wild type is non-pathogenic, but this rugose variant is pathogenic. So basically, after 24 hours, we were able to see pretty decent production of cyclic diGMP that's significantly different from the wild type. And then looking over a time course, at 48 hours, we were able to start to see a significantly different spatial distribution within this rugose variant. So as you can see, the cyclic diGMP seems to be kind of nearing the edge of the microbial colonies. Once again, a significantly different amount than the smooth wild type. And then after 96 hours, we can see this wild type start to produce a bit more cyclic diGMP, still significantly less than this rugose variant. So overall, I just want to highlight once again that we developed a method to directly detect cyclic diGMP in microbial colonies. And it's really important to visualize the spatial distribution of this compound because it can inform genetic regulation of biofilm production as it relates to virulence. And we're also currently using this method to visualize spatial distribution of cyclic diGMP in different Vibrio mutants. So keep your eye out for that one as well. With that being said, I will move forward onto the untargeted metabolomics part of my project. Once again, bringing this image back up, biofilm formation is very important for Vibrio fischeri. So with that being said, we use two different mutants of Vibrio fischeri to study this host colonization. So this first one is the biofilm down mutant. As you can see, it does not form a wrinkly phenotype on agar, and it actually doesn't even colonize the squid at all. This biofilm up mutant forms this really nice wrinkly phenotype on agar, produces a lot of biofilm, and it actually even outcompetes the wild type Vibrio fischeri to colonize the light organ of the squid. So everything that we are going to be looking for is going to be in this biofilm up mutant. And our main question is what small molecules contribute to squid colonization and biofilm formation. So in order to study that, we have a general workflow for untargeted metabolomics that involves large-scale culture, either in liquid or on solid media, some sort of solvent extraction, LCMS analysis with MSMS fragmentation, and then Laura talked a lot about many different types of data analysis softwares that we can use to identify features and understand differences in different biological conditions. So the portion of this that we wanted to begin to optimize was actually the first step, the large-scale culturing. So in these next few experiments I'll be talking about, these are all 1 liter cultures of Vibrio fischeri with X8016 resin. And we were comparing the biofilm up mutant to the biofilm down mutant at both 1 day and 6 days of growth. So this is a PCA plot showing that there is a unique statistical difference between our biological conditions as well as our different growth conditions. So these blue ones are the biofilm up, the pink is biofilm down, and then the X's are day 6 and the circles are day 1. So we see that all 4 of these biological conditions cluster separately in this PCA plot, which is really nice. So just looking at things at a very basic level, we do see that these data sets are statistically unique. So our big question next was we wanted to know what are the features that are making these data sets unique. So looking at the LCMS data, we do see between day 1, 4, and 6 for these biofilm up cultures that there is a nice increase in the number of peaks that we can see the bacteria producing over time. So if we just look in this region, this is reverse phase. So in kind of the more polar region, we even see lots of different peaks being produced between 1, 4, and 6 days, as well as kind of the middle region of this base peak chromatogram, and especially towards the end in the less polar region, we see a significant increase in lots of different features produced over time. So overall growth time is a really important component to the metabolite production in these V. fischeri cultures. This is just an image of my full GNPS network. We kind of wanted to dive into we saw an increase in the number of features, but we wanted to know what these features are. And so we decided to go with GNPS. Like Laura said, there's a lot of data in these GNPS networks. So although I'm not going to go into the details of this full network, we wanted to understand how to prioritize this data. So we wanted to look at clusters that did not have any annotations, that were only from the 6-day cultures and only in the biofilm up cultures as well. So with that, we found one unannotated GNPS cluster that only or mostly appeared in the biofilm up mutant of Vibrio fischeri, and it actually only appeared in day 6 cultures. So in this cluster, there's basically 4 different members, 276, 304, 290, and we wanted to check into the raw data to ensure that this also translated there as well. So this is an extracted ion chromatogram for M/Z 276. We can see that it only appeared in our biofilm up conditions on day 6. And then looking at the rest of the features within this cluster, we saw the same thing. So here is 290, and here is 304 as well. The intensity is pretty decent. We were also able to get decent MSMS data, which I'll show here. So this is the MSMS data for 276, and this is 304, sorry, and this is 276. And we saw that there is this shared moiety between these 2 compounds. So it's clear that they're related. we did some database searching just at the MS1 level, as well as looking more into GNPS, and we're unable to get any sort of database hits for these compounds, which is, exciting, but also makes our lives a bit more difficult. So currently, I'm working on isolating these features of interest for structure elucidation. I'm actively growing several liters of this bacteria, and the compounds also appear to be quite stable, which is nice to work with. And then they're also not in the growth media. And then moving forward, the big picture is we want to understand how these compounds contribute to biofilm formation, bioluminescence, and host colonization within this symbiotic system. So with that, I'd like to thank my lab and the NSF for funding, as well as the UCSC Department of Chemistry and Biochemistry. So with that, I'd like to take any questions.
Okay, thanks a lot, Allyson, for this very nice presentation. I don't know if some people have some questions. Maybe you can put up your hand or ask the question in the chat. Meanwhile, I have a question about when you are looking to the chemical diversity, you check at day 1, 4, 6. Do you know if at day 6 it reaches up the stationary phase of growth phase of the for the bacteria? And is there some specific signals that you can only get at day 1? Because that's something that's at a very, very early stage is not well studied, and actually there is some compounds sometimes quite unusual that you can find. Unfortunately, nobody look at them yet.
Yeah. To your first question, we actually paired this data with the colony forming units over time. And we did see that, I believe after day like 3 or 4, it seemed that the bacteria did reach stationary phase. We don't have a plate reader in our lab, so we weren't using that. But as for looking at the day 1 data, I actually haven't really dug into that as much, just because we know that natural products are typically produced towards the stationary phase of the bacterial growth cycle. And that's like mostly what we're interested in. But it would be interesting to see if there's, anything that is produced in day 1, that might be playing a role in the symbiotic system. So definitely something I'll have to look into.
Okay, I don't know if someone has some other questions. I don't know. Yes, I think one. Does anyone? I saw a hand, but no. And any? Okay. Maybe you can write it down. If you have a question, we can wait a little for your question. And do you have any clues about those compounds? If they have any matches, the few compounds that you highlight are specific to day 6, about what they are, or?
Yeah, I'm not sure yet. Laura seems to think that they might be lipophilic of some sort. But yeah, I'm honestly not sure. They retain to the column really well. So could be anything, honestly.
Okay. So there is someone is asking about the time that it takes for the work you are presenting today. How long does it take to get such results?
That is a wonderful question. The first part was a bit quicker. I think I did all of that work within probably 6 to 8 months, except for the writing, obviously. And then this second part I've been working on for the past I would say year and a half. I've been focusing on the untargeted metabolomics portion of this project.
Okay, thanks. So it's clearly I highlight how it's take a long time to get all the data first.
Yeah, definitely.
Thanks a lot, Allyson, for such a nice presentation. And I think we'll switch to the presentation of Karin Meyer, which is a postdoctoral researcher in Hoffmann-La Roche laboratory. And actually, we will switch from natural product and marine organism to actually the microbes that we can find in the digestive tracts and its impact on, I think, mouse health. So, Karin, the floor is yours, and thanks a lot for being here for this presentation.
Thank you. Actually, I'm not presenter. I can. Oh, super. Ha, that timing is impeccable. Thank you so much. So you can see my screen now. Super. Yes, exactly. I'm currently a postdoc at Roche, but I actually will be talking about the project that I worked on during my PhD, which is, as you very professionally introduced, related to the gut microbiome. So in this project that we've termed Mouse Metabolic Landscape, we were interested in understanding the gut microbiome. And a lot of what is known about the gut microbiome relates to the physiological variation along the length of the gut. So there's gradients of oxygen, nutrients, pH, and well, everything that changes along the length of the gut, going from, for example, the small intestine to the large intestine. And what comes with that is considerable variation with respect to the community composition. So what I've depicted here in a bit of a higher level overview is the difference of microbiome composition from the small to the large intestine, and the differences between the mucus that lines the intestinal tract and the intestinal contents that move through the gut. And this is based on 16S sequencing data. And already on that level, we can see large differences. And a lot of these microbiome factors are pretty well studied, actually. But what is much less well understood is how metabolism changes over the length of the gut. And that has implication for a lot of factors. So understanding how does metabolism change and which metabolic niches exist in the different areas, how are those specific and what are they characterized by, can help us understand what the microbes encounter that see these niches, and that this foundational research has implications for infectious diseases, drug metabolism. A lot of other fields can rely on that information. So we started out with the basic question, which metabolic niches do we have in the gut? And since gut samples are a rather complex type of samples, we developed an LC-MS method to measure that. And so we started out by identifying all of the molecules that are relevant in the gut. And this is what is depicted in this graph here in the gray bars. And this annotation or classification of compounds is based on biopsych. And ultimately, then we try to find and order all of the standards that would help us cover this basis, the chemical spectrum. And what we ended up establishing is a quantitative LC-MS method that focuses on 128 gut relevant compounds depicted in blue here, covering all of these classes. So with this method, we are able to deal with the high salt concentrations that we have in the complex matrix of the gut. We annotate with a higher confidence, and we're able to resolve isomers, which is especially important with respect to bile acids, which are highly important in the gut microbiome. And with this method, ultimately, we mapped out the intestinal metabolism of mice for more than 400 samples. And when I say map, I keep saying map a lot. I do realize that we really mean we took the entire intestinal tract of the mice and chopped it up in 15 pieces, which is what you can basically see along the x-axis here. So that's one site in the stomach, 10 in the small intestine, and 5 in the large intestine. And then we measure all of the metabolites there and map out quantitatively what we can measure. This is obviously a little cropped in this heat map here, what you can see on what would be the y-axis, and the color code represents then the different molecular classes as defined in biopsych. And just on a very high level, you can see that this is a clustered heat map, and the color code, blue means low, red means high. We can see here that, for example, there's a very clear signature for the stomach microbiome or metabolome, where specific molecules make up the stomach. And overall, we can see a shift from the small to the large intestine, again, based on clustering, where the small intestine is mainly dominated by amino acids, whereas in the large intestine, we see a lot of fermentation products, small organic acids, but also vitamins, which we found pretty interesting at that level. And ultimately, with this metabolic map and this high resolution, what we can now do is draw spatial profiles. And so what you can see in this plot here is the concentration of any given molecule. Here we've chosen allantoin over the length of the gut. So these are the 15 sampling sites. And we can draw these spatial profiles here where we see that, for example, in these so-called specifically pathogen-free mice, the fully colonized mice, we see this sharp increase of allantoin concentration at the end of the small intestine. And this first and foremost is not usually seen because most intestinal studies for ease of work focus on feces or just pool the entire intestinal sample. And so it really requires this level of resolution to see sharp increases and distinct profiles such as this one. And now when we combine that with information from germ-free mice, so these mice are void of any bacteria and serve as amazing controls for essentially what mouse metabolism, intestinal metabolism looks like without any microbes. And in that comparison now, where allantoin, in this case, concentration doesn't change at all over the length of the gut, we can now speculate why we see these differences. And I wanted to, with this data where before, based on sequencing data, we always have the information of which bacteria are present in the different sites. And now we get the functional information saying, what are they doing? What is happening there? For example, something with allantoin. And I already alluded to the fact that we can now speculate about what is happening there based on a microbial activity level. And for that, I pulled up another example, which is short-chain fatty acids, in this case, butyrate and isobutyrate, where here in the cluster of the large intestine, we can see a sharp increase expectedly in the large intestine. And then when we correlate that with 16S sequencing data, which we also obtained in a matched experiment on all of these different sites, we can now see which microbes coincide with the increase in concentration for specific metabolites, and ultimately come up with hypotheses about what is going on here. This is obviously something we would want to see, but for new molecules, or not necessarily new, but molecules for which there weren't explanations based on the microbiome, we can now come up with ideas to test further in the lab. And with that, we now have a full map of intestinal metabolism, which is able to reveal these distinct niches in the small intestine and the large intestine. What I entirely neglected in this talk now for time reasons is that we actually distinguish between the mucus samples as well as the content samples. And there's a very clear difference, not just with respect to community composition, but also metabolism changes when looking into the mucus environment. And ultimately, using the different data that we've obtained, including 16S sequencing data, we can now come up with these metabolite microbe associations and connect which microbes occur where with those bioactive compounds, and ultimately, have ideas about who does what, essentially. And it's a rather large paper, so I had to cut it short, and I would like to stop here, thanking all of the collaborators that helped bring this project to paper, essentially. And thank you for your attention, and I'm happy to take any questions.
Thank you very much, Karen. It was a very nice presentation. I see that we already have a first question in the chat from our next speaker, Thomas Carton. So Thomas is asking, how much sample do you need to analyze all 128 metabolites?
You mean material? It's actually not a lot. You'd be surprised how little material you need. I think it was a few milligrams actually per site, because especially with this resolution, it's a bit of a problem because obviously in the small intestine, there isn't a whole lot of content, especially if you cut it in this. So we really wanted to go for the systematic approach. Ultimately, I'm going to spoiler my paper a little bit. Ultimately, you can actually conclude that you obviously don't need it for all of the metabolites. For a lot of them, it's totally fine to just say, okay, small intestine, or then distinguish in duodenum and yeah, those rather large classifications. And we had milligrams of samples, really not a lot.
Okay, thanks.
Thank you.
Thanks for the question.
Samuel, do you have any question, or can I go ahead? You don't seem to. Your mic is muted.
Yes, and do you know, have you seen any regulation like metabolite from the mouse itself that can regulate the bacteria? Because you have a lot of metabolites. Some you know can come from the bacteria, and some can come from the mice as well. Do you know the proportion, and if there is some back control using those metabolites?
So this is a very complicated topic, and we struggled with this quite a bit because obviously there's a bunch of metabolites that can come from a bunch of different sources. And what I think I had it on one of the slides, we actually measured the diet as well. So we really just took the food pellets and extracted that to get a feeling for what is just diet metabolites, and then try to understand a little what comes from where essentially. And there's always a few, let's say, positive controls like primary bile acids, for example. So those are obviously mouse related, and these we see beautifully. You see them exactly where you should see them at the beginning of the small intestine, when then they're being reabsorbed or altered into secondary bile acids. And the interaction with the gut microbiome, we did see this with the tryptophan metabolism or tryptophan metabolites. There's a few that are going through I think hepatic uptake, and then they're being changed, especially all of the indole. I think indoxyl sulfate is the one, maybe. So one of them is actually taken up by the mouse and then changed and then secreted again into the gut. So we can see that. But one of the problems is obviously that this is not dynamic. We just have one snapshot, and then we try to basically come up with, yeah, where does it come from? Why do we see it? But yeah, it would obviously be very beautiful to follow that on a time aspect.
And
thank you.
I also have a quick question about your more genomics data with your metabolomics data. So you rather done correlation, and I was wondering, because I went to check the paper and went through it so quickly. Did you were you plan or did you thought about doing more integration, data integration, like fuse the two data sets through multi-block integration, for example, and to try to have a stronger statistical performance of your model? And if not, why is it a choice? Or?
Yeah, I remember at the time we went through our options of how we can, because we kind of look at the basically metabolic potential of the microbes. So we use the 16S data to inform about what is metabolically possible, and then infer based on like a protein level to a level of understanding what can be produced from the microbiome. So it's true that we do not brute force correlate and integrate that data. And at the time, we chose not to because the data was rather noisy on all ends. And when I said we have it matched on a level where we say we have one sample for the duodenum, for the ileum, for the ileum, for the cecum and colon in the intestine, but we do not have the exact same resolution because that's kind of coming back to Thomas's question before. We just do not have enough material to get enough DNA out of it for 16S sequencing. So we were a bit worried about how meaningful it would be at the end based on from a statistical point of view, because the data is rather noisy and we would probably we were worried that it's a lot of work for not a lot of information, but we have thought about it at the time. That is true.
Okay, great. Thank you very much.
You.
It was very clear.
Thanks.
If there is no other question in chat, so it doesn't seem to. So thank you again very much, Karen, for accepting our invitation and for this nice talk. And we'll be now going to the last speaker of today's webinar. So it's my pleasure to welcome Thomas Carton, who currently serves as a scientific operations manager, working at the intersection of science, clinical and lab operation, research and development, and customer support in the analytical field. So please, Thomas, the floor is yours.
Thank you. So thank you for inviting me to give this presentation on the contribution of mass spectrometry in clinical studies with two practical cases. So clearly it's a story about metabolomics, but told by a molecular biologist. So sorry for that. First of all, a short presentation where I want to talk first about my lab Biofortis, of course, then introduce how we consider omics for the microbiome, before giving you the two examples of what we can and why we need this technology within our study. So Biofortis, we are a contract research organization. We provide services for all stages of clinical trials, and we are currently based in Saint-Herblain, near Nantes, in Paris, and in Berlin. And our core business is to conduct clinical trials that enable us to prove the supposed effect of an active product. So we propose clinical operations, biometrics, data science, consulting, and we propose also laboratory analysis. And just one focus regarding the analysis. We started working on the microbiome maybe 17 years ago, and we currently have maybe the most complete next generation sequencing, so metagenomic platform in the Nantes area. So just some words about microbiome and omics. When we start working on the microbiome 17 years ago, we started working on the microbiome, considering it mainly as an ecosystem composed of several different species of microorganisms, bacteria, archaea, and so on, with many active products that can or should lead to changes within the ecosystem. It can be a probiotic, it can be prebiotic, it can be drugs as antibiotic, and so on. And of course, in the recent years, all the discoveries around the microbiome lead to no longer consider it as a simple ecosystem. If we take the example of the intestinal microbiome, the gut microbiome, we know better understand the power of the link that exists between the microbiome and us. So the gut microbiome is involved in very essential functions within the human body, metabolic functions, protective functions, more interestingly, trophic functions. By using animal model without the microbiome, we discovered that without the microbiome, the body cannot develop in the same way as with the microbiome. And because the microbiome is involved in many essential functions, we need to use some other methods in order to consider not the microbiome as a simple ecosystem, but the microbiome as a part of an holobiont. So the last year, the study of the microbiome has evolved mainly thanks to the metagenomics tools. targeted metagenomics or metabarcoding, as presented in the previous presentation. Now we prefer to use the true metagenomics, but considering not the microbiome, but the holobiont, requires extending the field of investigation, and we need to add some other methodologies as metabolomics, proteomics, lipidomics, and some other very strange omics method. Just it looks very simple on the previous slide, but I have an important point to remember because we are working on clinical trial. And in a clinical trial, an analysis necessarily belongs to one of those categories ranked by a decreasing order, decreasing importance. An analysis in a clinical trial is either a screening criteria or the primary endpoint, or a secondary endpoint, or sometimes an exploratory endpoint. And obviously, the importance is correlated with the level of quality of the analysis that you must have within your project, within your trial. And this is one of the reasons why it is often preferable to choose targeted methods, and particularly in metabolomics. So just two cases I want to talk about within this presentation. The first one is about short-chain fatty acids, so very small molecules from C1 to C5, of which the three mains are acetate, propionate, and butyrate. We discovered a lot about the short-chain fatty acids, and from the end of the 70s up to the 90s, we found that they are produced mainly by the microbiome from the fibers, from the dietary fibers, and that they act on a wide range of host mechanisms. It can be cell metabolisms, it can be apoptosis, immune regulation, endocrine functions, and so on. Within this first project, it was a preclinical study. The aim was to prove the prebiotic property of one fiber on a piglet model. So one fecal sample was taken before consuming the fiber, and one other sample after the consumption, and one then analyzed first to see the impact on the ecosystem. And it works. The fiber lead to an increase of the bifidobacteria population within the microbiome. So we proved the prebiotic effect on the ecosystem. But of course, we wanted to go further and see the possible impact on the host for the microbiota. So the way we did it was to prove by mass spectrometry that fiber intake lead to an increase of the short-chain fatty acids. And it was the case for two acetate and butyrate. So within this first case, by combining these two approaches, we could consolidate the evidence of efficacy of the product on the host for the microbiota. For the second case, it concerns a large family of biliary acids. So within the bile acids, there are a lot of different molecules. Primary acids are produced by the human body, and many of the secondary bile acids result from the metabolisms of the primary acids by the microbiome. The second project was on very interesting molecules, because what we have learned about these bile acids over the last few years is that they interact directly with the host immune system and through the bile acid receptors. And more interestingly, the secondary acids produced by the microbiome have more affinity, as you can see here, with the receptors of the immune cells than our primary acids. So in these mechanisms, it means that the microbiome is more efficient than we are in activating our own cells. So the second project, it's a clinical trial. The goal was to completely replace the microbiome in immunocompromised patients, very important, having a dysbiotic microbiome. So I mean a very disturbed microbiome. This replacement is done by a fecal microbiota transplantation, an FMT, whose principal and main way of administrations are described on the right side of the slide. Currently, the FMT is already validated for the treatment of the C. difficile infections and is under investigation for a lot of different pathologies from ulcerative colitis to obesity, Parkinson's disease, autism, and so on. So within this project, the aim was to replace the microbiome in immunocompromised patients. Obviously, replacing the whole ecosystem, the microbiome, results in a change in all the ecosystem. And I'm not showing those results because I just want to focus on the metabolite side. First of all, metabolomics allow us to observe an increase in the concentration of the three main short-chain fatty acids before the FMT, before the fecal microbiota transplantation, and after the FMT, you can see an increase of the three main short-chain fatty acids. But more interestingly, in these immunocompromised patients, FMT also allows us to restore certain mechanisms of activation of the immune system. And in particular, with the return to the production of the secondary bile acids that were highly depleted before the FMT, and that increases a lot after the fecal microbiota transplantation. So again, this is a combination of the analytical approaches that allow us to better understand the effectiveness of a treatment, here a complete transplant of the microbiota. So that's all from my side. As a take-home message, I just want to highlight that if simple omics approaches can easily explore the ecosystem, this famous ecosystem that is the microbiota, only combined approaches truly allow us to understand how we can act on the holobiont. But I note that at the beginning, Laura said exactly metabolomics, genomics, one combined are greater than the sum of their parts. Totally agree with her. I think I made the cycle. Thanks for your attention.
Thanks a lot, Thomas, for your very nice presentation. If some people have some questions, please don't hesitate to ask on the chat or to ask directly. I was wondering, because actually you focus rapidly on targeted metabolomics.
Yeah.
It's because your aim is to be sure of the biomarker you are following, or why is that choice in particular?
Because in a clinical trial, the number of volunteers you include within the trial is determined by your statistical calculation in order to reach enough statistical power. To do that, you need to perfectly know the variability of your methods. You need to know the expected variability induced by the treatment. And it's the reason why you only can calculate the number of the volunteers based on a very small number of metabolite. You can do it on short-chain fatty acid. You can do it of 1, 2, or maybe 3 or 4 different bile acids, but you cannot calculate the number of volunteers if you want to use untargeted metabolomics as a primary criteria. It's not possible. And if you cannot prove and describe the way you determine the number of volunteers you have to include within your trial, the ethical committee do not validate your protocol.
Okay.
It was actually my question, Samuel. You took it away from me. But then I have another question. Then how do you choose the metabolites that you're going to be targeted? It's known biomarkers already, or is it because the untargeted study could help you to actually choose the biomarkers that you will be targeted after in your following study?
Yes, you're right. Of course, by using untargeted metabolomics, you can identify new biomarkers, new molecules that could be of interest for some applications. We are a contract research organization conducting clinical trial in order to prove the efficacy of some product. We use all the discoveries you can make within your own laboratory by using untargeted metabolomics, but we need to focus on the metabolite that can be related to the product we want to test. It's clearly a clinical trial related to the efficacy, the expected efficacy of our impact of the product, of the treatment. So when you write your clinical trial protocol, you need to write a rationale, a scientific rationale, where you explain why you choose one metabolite and not one other. So of course, within the scientific rationale, we use all the papers published by all the scientists that use untargeted metabolomics and who find in the past the short chain fatty acid, more recently the bile acids, and maybe tomorrow TMAO or these other metabolites we are talking about regarding the microbiome.
Okay. And I actually have another question, but a quick question, and maybe I missed it afterwards, but you say it was important that your patient has to be suppressed. Is that right?
Yeah.
And why that exactly? Or maybe I missed the full explanation.
Also, it's quite simple. It's because they are immunocompromised that the bile acids became very important to observe.
Okay.
And that's all.
Okay. Thanks.
Okay, so I think we are nearly up to the end. Thanks a lot for your presentation, and I would like to thank all the speakers today for their presentation around all the Holobiont concept and how how Metabolomics is able to study such complex system. We have a few announcements to tell before the end of this session. So the first one is that the RFMF is going to propose 4 new webinars next year. So stay tuned, and we will provide you the information about when they will be organized and on which thematics we will organize them. You may have seen also that we are providing all the video for the webinars directly on the RFMF website, and we are trying to see how to provide them on YouTube, and also to provide them as a short video that everybody can visualize and check if they want, and if they want to go back to some older webinar. And one last information for those who might not have the information yet is that the 70th scientific day of the RFMF are planned to be between the 10th and the 13th of June 2025 in Paris. Stay tuned to all the information about the registration. It should open at the beginning of February, and the closure of the early bird registration should be around the end of March. So that's all for all the information. Thanks a lot for everybody to have to be there for this webinar. I hope this one was of interest for you. For me, it was very, very interesting. Anyway, and again, all the presenters, thanks a lot for all your nice work on all the work you have presented today.
Thank you very much, everyone.
Thank you too.
Bye.
So I think we can now, yes, close up the session, and I think